Hello viewers, welcome to the introductory plant pathology, and today we will be dealing with the diagnostic characters of the important genera of fungi. If we compare the diagnosis of the disease in plants with the human beings, we see that when a patient goes to a doctor, he observes all the things from of the body, and he asks about the history of the disease. He takes the pulse, he sees the heartbeat, and so on and so forth. Similarly, in the plant systems, when we see a diseased plant, it is very necessary to find out that what type of pathogen is infecting the plant, and we can say that what type of disease is affecting the plant health. Therefore, diagnostic characters of fungi is very important to know. The first step in managing any of the several diseases caused by fungal pathogens is to obtain an accurate diagnosis. There are various steps and activities which are associated with the accuracy of plant disease diagnosis. If we discuss the different aspects of diagnosis in the plants, we see that first step is the identify proper plant or host. We should know about the plant which is diseased, because by knowing the plants we can find out that what type of diseases are affecting this plant. We should also consult the compendium of diseases, so many list of the pathogens available associated with the disease of that particular plant, and therefore we should see the healthy plant as well as the diseased plants. Otherwise, we will not be able to compare and to observe or examine that what type of changes have been occurred in a diseased plant which are not there in the healthy one. Then recognize healthy plant appearance. So, the appearance of healthy plant is first examined, and then we compare it with the diseased one, that what type of above ground parts are affected, whether below ground part or root parts are affected, leaves, flowers, stems, like these parts we should examine completely. Then we observe the symptoms and sign on the body of the plant. So, symptoms are very important for finding out the causal organism affecting the plant health. These are the characteristic symptoms. Characteristic symptoms of the plant disease are mostly stunting of the plants, where the plants is stunted, internodes become shorter, and inadequate development of root, malformation of leaves. There are certain puckering and so many deformation in the leaves occur during diseased conditions. And inadequate chlorophyll production we see in so many plants when they are affected by different pathogens. The chlorosis is the main symptom observed, because affected plants show the deficiency of chlorophyll, or because of the disease chlorophyll is not formed in the affected plants. Failure of flowers and fruits to develop, and similarly we see that in the diseased plants flowers do not develop. If at all the flowers develop, the fruits will not be formed. Thirdly, the wilts or dieback type of symptoms can be observed. Shoot or leaf blight can be observed. Leaf spots and fruit rots type of symptoms can be observed. We know that there is a disease triangle. For developing a disease, three factors are very important. First is the susceptible host. If our host is not susceptible, the disease will not occur. Then there should be a favorable environment. Disease appears only when the environment is favorable for its development, and then the virulent pathogen. The pathogen which is having the capacity to infect the plant must be very virulent, otherwise the disease will not appear. So, this is a disease triangle which shows that these three things, these three factors, environment, plant and the pathogen, they are in a equilibrium position and they help in developing a disease. First of all, we take the Phytophthora as a causal fungus causing different types of plant diseases. So, the pathogen Phytophthora is commonly referred to as water molds. They thrive, grow, reproduce and infect plant roots in water, saturated soil and along river banks and ponds. Flooded or saturated soils are not favorable to plant growth and can predispose plant to infection. And third, excess water or poor soil drainage make roots vulnerable to the pathogen. By this way, we learned that for Phytophthora, actually soil drainage should be very important point. Water should not be there near the plant, and especially it is a pathogen of nursery diseases or the orchard diseases. If we see the classification of the Phytophthora, we see that it belongs to the class Oomycetes, order Peronosporales, family is the Pitheaceae and genus is Phytophthora. Phytophthora causes foot rot, root rot, crown rot, gummosis, leaf fall and brown rot disease in well grown orchards. Here I am showing you the damping of symptoms. You are seeing here the seedlings of the plants showing the rapid death and collapse. Seedling appears broken at the soil line as is visible in the photograph. Then the drying branches of trees. Phytophthora is a very characteristic pathogen and it affects big trees in a way that they suddenly dry and a side of the tree show the drying. So, the sudden drying of branches, especially on one side of tree, is the symptom associated with the root damage, and the disease is suspected caused by the Phytophthora as seen in the film. These are the sporangia, the spores responsible for causing disease. Then the zoospore, here it is the zoospores, this is the sporangia, this is the chlamydospore and this is the matured oospore prepared for giving out the sporangiospores. Then we will discuss about the factors which favor the disease development of Phytophthora. Phytophthora parasitica and Phytophthora citrophthora are the species of common occurrence on citrus in India. This can tolerate temperature up to 35 degrees centigrade and thus is most active in summer and forms chlamydospores and oospores for its survival in winter. Thus, Phytophthora remains active throughout the year in nurseries and in irrigated orchards. This is the life cycle of the Phytophthora infestans, where we see that oospores remain always in the soil and on the plant debris in the farm. They germinate to give sporangium, and sporangium when germinate they directly give the germ tube, or they produce swarming zoospores with flagella, and they in turn infect the plant. This is the leaf section of the plant showing the sporangium coming out from the stomata, and this is how again it infects the plant in a recurrent cycle and completes it in a asexual way. In sexual way, the sexual reproduction, oogonium and antheridium are fused using the fertilization tube, and after fertilization the thick walled oospores are produced, which after reduction division gives rise to the sporangium, and sporangia in turn again infect the plant. This is how Phytophthora infestans completes its life cycle. Then the second most important genus we are taking is the Peronospora. Peronospora is also causing very destructive diseases, especially the downy mildew, and it is a obligate parasite. It causes serious foliar diseases on many host plants. Here we see the downy mildew in broccoli caused by Peronospora parasitica. You see here that white downy growth are seen under the leaf surfaces, and branched sporangia force are there. In this case, you see here it is branched, and it is produced single sporangium on the tip, which emerges out from stomata as shown in case of Phytophthora. It infects thoria, yellow and brown sarso, rose and cauliflower. They are very economically important plants. So, this is the life cycle of Peronospora. Infection arises initially from the oospore. It germinates in the soil, and plants are colonized by growing mycelium. Conidiophores bearing asexual, spherical, hyaline conidiospores, and these conidia grow out of stomata. Here you see it is growing out of stomata. On germination, conidia initiates new rounds of infection, and this type of asexual reproduction is seen. Then in the sexual reproduction, oospores are formed concurrently, and the female oogonia is again fertilized via a fertilization tube from the male antheridia here. The fertilized oospore develops, and oospores are profuse on infected leaves, which again infect the plant. This is how the Peronospora completes its life cycle in the plant. Then we come to the another most dreaded disease caused by Sclerospora. Here we will be talking about the Sclerospora graminicola, which is causing downy mildew disease in bajra or pearl millet. Here two types of symptoms are produced. One is the downy mildew symptoms, and another is the green ear symptom. When we grow the plants in the field, when we sow the seeds, after 2 or 3 days we see that plants are showing the signs of chlorosis or yellowing. This shows that Sclerospora graminicola, the causal fungus of downy mildew, has attacked the plant in the field. The affected plants, when they grow again, they show stunting, that is the shortening of the plants in comparison to their healthy counterparts, and number of leaves and nodes are reduced, resulting in reduced yields. Disease appears in the form of chlorosis at the base of infected leaves, followed by sporulation on the lower side of leaves known as the half leaf symptom. This half leaf symptom is the characteristic feature of the downy mildew in pearl millet. At the time of panicle emergence, green ear symptoms become visible. Symptoms appear on ear head with all possible degrees of proliferations and malformations. In malformation, the florets are converted into leafy structures of diverse appearance. Asexual sporangia and zoospores are produced, and normal temperature 20 to 25 degrees centigrade and high relative humidity, that is 95 to 100%, favor the sporangial production. This is the picture showing the downy mildew disease of pearl millet. This is the characteristic half leaf symptom, where the half below the leaf is showing the downy growth having the sporangia completely filled within it. And this is the types of malformation. This is the healthy ear head in the extreme left, and these all types of malformations we are seeing here, in which the lower half is having green ear. This is the completely malformed ear head. Then this is the tufting, branching, layering, and so many types of Frenching. Different types of symptoms are seen in this picture. Before going to the basidiomycetes or the smuts, I would like that we should understand the generalized nuclear cycle of the fungi. The generalized nuclear cycle of the fungi show that there are gametes or gametic nuclei is n number. It is a monokaryon. Then there is a plasmogamy. Then 2n dikaryon, then karyogamy occurs, a zygote, 2n zygote that is diploid in nature is formed, and after meiosis or reduction division, haploid spores or gametes are formed. So, this type of general life cycle is there in the higher basidiomycetes or smuts and rusts. So, we first take the astilago, the smut fungus, and we will be discussing here about the wheat disease causing loose smut of wheat. Here you are seeing that this is the healthy panicle of the ear head having different florets of the flowers, and here you see that at the place of grain formation you are seeing only the sooty mold like black powder which is being done by the astilago fungus. This plant is systemically infected, looks normal till ears appear. As soon as the ears appear, we see the sooty mold on the ear heads and there is no grain formation at all. So, this is economically very important disease. Seeds covered with thin layer here and which rupture to disperse in a black sooty mass, they are telutospores. And in case of the corn, where the astilago maydis is there, the fungus forms the galls. You see here the big gall caused by the smut disease on the above ground parts. This is the life cycle of loose smut of wheat. This is caused by astilago nuda, Forma specialis tritici. Here we see that mycelium invades the, actually it is the seed borne disease and systemic in nature, as I told you earlier. The mycelium invades part of the embryo in the seed. This embryo develops with the germinating seeds, and the mycelium follows growing point of the wheat plant. Mycelium invades the grain sites and the whole panicle appears as a sooty mold. Grain site replaced by masses of teleospores, and teleospores land on flowers of healthy plants and infect the developing grains in the healthy plants, and like this whole life cycle of the loose smut of wheat is completed. If we see the classification, we see that phylum Basidiomycota, class Astilaginomycetes, subclass Astilaginomycetidae, order Astilaginales, family Astilaginaceae, and genus Astilago, and species which is infecting the wheat is Astilago nuda. This is the Sphaeculotheca or the covered kernel smut. In this, teliospores of sorghum Sphaeculotheca sorghum are seed borne and germinate within the seed. Seedling plants are infected, but symptoms generally are not apparent until heading. Smutted ovaries are covered by a thick membrane, and seedling infection by sporidia produced by teliospores present on the seed or in the soil. Here we see that this is the healthy panicle of the sorghum, and you see the affected portion completely black by the mold. Then we come to the Melamsora or the flax rust. Flax is a very economically important plant, oil yielding plant. This shows the small yellow pustules bearing spermagonia or upper side of the young leaves. Orange or yellow acial appear on lower surface of the seeds. Iridophora on leaves and stem also, you see the stems is also infected. Rust having sessile, so there is no pedicel under the spores, they are sessile. One celled teliospores, these are the teliospores, and they appear in a single layer. Then we come to the alternaria leaf spot. This I have taken as a representative of the family Deuteromycetes or Fungi imperfecti. Otherwise, there are large number of pathogens in Fungi imperfecti which are causing diseases of economic importance in the plants and crops. Here I am taking only the alternaria as a leaf spot. It is the most characteristic symptom is brown or dark spot on older leaves, and you see the circular or angular leaf spots enlarge and soon develop dark concentric rings and ridges, giving a target like appearance here. Spotted leaves soon turn yellow, wither and drop. Leaf spots are usually covered with dark velvety spores. We will find dark velvety spores of alternaria on these spots here. So, this is the leaf spots infected with the alternaria of guar crop. This is the sesamum plant showing the leaf spots on the fruits as well as on the stems and on the leaves. This is the causal organism alternaria. You see the conidia of the alternaria having the alternate septation, therefore, it is known as alternaria. And this is the simple life cycle of alternaria brown spot caused by alternaria alternata, and here you see the conidia on dead tissue and canopy in orchard remain in the field always. They are airborne in nature, they infect the young leaves and fruits and stems of citrus in this case and infect the plant, and again develop the leaf spots and fruit spots and on the stems brownish to blackish leaf spot of alternaria, which again give rise to the conidia, and this type of life cycle is completed, because we know that in Fungi imperfecti there is no sexual reproduction, there is only the asexual type of reproduction. This is the ergot of wheat caused by Claviceps purpurea, and you see this is the ergot sclerotia. They are pseudo masses of the fungus, and when they germinate they give rise to the ascospores. This is the cross section of the ascospores, and we see so many ascospores filled in this. Here it is, and when they are liberated through air, they again infect the healthy plants, and they infect again the healthy crop in this way. Today we discussed about the diagnostic characters of the causal organism or the pathogen which are causing economically important plant diseases in crops and other plants in the orchards and nurseries. Most important features of diagnostic characters were discussed, and we have tried to learn the disease caused by Phytophthora, by Claviceps purpurea, by Alternaria, by smuts and rusts. Thank you.